indeed Tracy Williams is printing them. I asked Tracy Williams to print them, and there was a long pause. She said, all right. So, what I'm going to do is I'm going to run through some slides of the talk I gave recently, to kind of be something of a recap over what I did last week, with a slight twist. And I will pause in the middle to show, and then we will look at a different way of analysing genetic processes, which gives another kind of insight into genetic variety. So, the other thing I want to do is to say your coursework, which I'm not going to do today, but I'm going to, what I would like to do is, during this week, I will set you the coursework, which will be in 2 parts. The total coursework will be worth somewhat less marks than Mark Helster's coursework. I think his coursework will count, as you've already done, will count for 2/3 of the coursework marks, and mine will count for 1/3 of the coursework marks for this course. And I want to set it for it to be, well, I'll take a vote. I can either set it for it to be due at the end of this term, in which case there'll be less of it, or I can set it that it's due when you come back next term. And so you have it over the holidays, which means, of course, you can procrastinate now. It's great. I'm relying on procrastination. I much prefer to do it this way. And the course will be, I'll supply basic code in 2 parts. One of them is a set of instructive fitness functions to do a genetic algorithm for, and you will see that dreadfully they work for quite reasonable fitness functions. And the second one, I'm going to get you, that's for traditional genetic algorithms. In the second one, I will put together, I'm going to do some, I would like to do some distributed research. That is, these algorithms I've been describing to you, as far as I know, they're new. I haven't seen them. And so what I'd like to do is to draw up a list of simple fitness functions, and let each person take a fitness function, or you have a choice of fitness functions, and run these algorithms and see how they work. And then, so you run the algorithm, you see what the results are, and comment on it, which is something you can put as, which you can take quite, it's quite a small amount of work if you run the algorithm. You can get some results, and you write a few lines of comments, or you could do really quite a lot of work on it if you felt like it. I'm also interested in, I think I'm going to be in supervising projects on this, if anyone's interested in the spring. When do you have your project meeting to choose projects?
Sometime in January.
I think it's sometime in January, yeah. It was last year. So you'll be fresh from this coursework and say, right, well, thank God I've done that, and then we can look at that again. Oh, sometimes you might say, well, that's kind of cool. So let's, no, we have one of those kind of ape intelligence puzzles. It's very sad. Somebody obviously lost it with the string hanging down over the blackboard, and I don't think I can reach that even if I stand on a chair. So I need a hook of some kind. Is there a tool? I'm searching for a tool in the manner of an ape in an intelligence test. I'm pretty sure I can't reach it on the chair. Book on the chair, stand on my lap.
Stand on the chair, why not?
Sorry?
Stand on the chair. I think you'll reach it if you stand on the chair.
Stand on the chair and use the chair.
So you can stand on me.
Yeah.
Well,
Just about.
Okay. Right, okay, finally. So, what am I going to do? I was rather hoping that much of what I was talking about last week was going to connect kind of tangibly with other stuff that you've been doing on the course. I mean, have you encountered Gibbs sampling on the course? No?
Yes. Some, but I think only some of us.
Only some of you, right. Okay, because it's only on one of the courses.
Yeah.
Okay, right, well that's so some of you actually won't have encountered it at all, so I do need to explain it for all the people who haven't seen it. Yeah, okay. Okay. Right. So, right. Right. So, as I was saying, my aim here is not to give you the structure of the field of genetic evolutionary computation. It's essentially a sort of an area of the wild west of science where lots and lots of people have had bright ideas, and if you find a bright idea that works for something, you carve out a little territory, you have your 10 acres and a mule at the scientific frontier, and you see if it prospers. Yes, I think I do want to stop Skype click to call. Right, and is it F5? Let's probably view full screen, isn't it? Right, so let's go through zoom to natural size, full screen mode, Control L. Right. As I pointed out, evolution is like a powerful learning algorithm. It's produced fantastically complicated things. It's remarkably fast. We know how it works. And genetic algorithms. So what we want to do, what I'm trying to do, is to find a model as simple as possible, which gives you essential aspects, and leaves out accidental aspects. And you only discover what's essential and what's accidental afterwards. Now, the basic complication, the reason for the entire academic industry on genetic algorithms is that natural evolution and indeed models, most of the models of artificial evolution are really hard to analyse. And the reason is that they're Markov chains, but they're not reversible Markov chains. Markov chains of a complicated type. And the reason for this is that you can breed children from parents, but you can't breed the parents back from the children. Why not? Well, these are sort of supposed to be parental genomes, and we can't. Every part of a child's genome is a copy of a parent's genome. It's a direct copy of a parent's genome. Right? You look very doubtful. No? Good. But by the pigeonhole principle, I mean, if some parents have, let's suppose the population stays the same size. So then we've got n parents and n children, the same amount of genetic material in the parents and children. then if some parents have more than one child, and some bits of parental genetic material are copied more than once, which we're assuming there's going to be selection. The selection is going to be random or systematic, but whether it's random or systematic, some bits of parental DNA are going to be copied more than child DNA. Then that must mean that some parts of parental DNA are not directly copied to children. So there's an inherent irreversibility in the process. If you have a DNA lab and you say, okay, we found the bones, we found 2 mass burials of a tribe. It was all parents and then all children. And so, but we've lost the labels. We don't know who are the parents and who are the children. Then you can find it out. You sequence all that DNA and you can see which is which. Now, if a Markov chain is irreversible, it's hard to analyze. You can't use any of the nice tools of statistical physics. In physics, you imagine physical systems as Markov chains as well. You have lots of gas particles going around, moving around, and bumping into each other. And in general, in physics, on the microscopic level, all the transitions, all the changes are reversible. And in physics, the probability of a microstate, or a probability of a state of your system, is proportional to the e to the minus energy of system over kt. And so you have some small system. You've got a spring of mass and another spring and another mass or whatever. This is my system, which can be in any of a number of states. It could be, say, 2 particles which are either sticking together or apart. Whatever the system is, you define a set of states of it, and under random thermal energy, everything's moving and bumping into each other, this will change state. But the fraction of time, it's the basic assumption in physics, the fraction of time this thing spends at thermal equilibrium, and it's in contact with a heat bath at temperature T. The basic assumption of thermodynamics is that the probability of states of each individual state of the system is proportional to E to the minus energy of the system over kT, and it's reversible. Well, this is a nice and simple Markov chain. So this is a nasty Markov chain which is considerably harder to analyze. So this, I think, is very convenient for large parts of the field of genetic algorithms, because it means that the problem has been set up in such a way that analysis is really hard. There is a book where somebody called Michael Vose, V-O-S-E, who tries to do the really hard analysis of simple genetic algorithms as a Markov chain population, transitions of populations, looking and saying, okay, let's take 1 point crossover and looking at the statistics of transitions. But it's really hard analysis. It's very complicated. So, I'm suggesting something, anyway, much simpler, which is to take, let's have a tiny genetic algorithm. And last week we discussed it with zeros and ones. This week I'm going to discuss a slightly different version of it, and more general, and considerably more applicable. One of the difficulties of applying genetic algorithms, and if you all zeros and ones, is how do you code everything out as zeros and ones? But of course, when you look at applications of genetic algorithms, almost nobody does. You find that everyone who applies a genetic algorithm, in fact, has used real numbers, and in fact, nearly everyone who applies a genetic algorithm has thought of their own genetic crossover operator as well, with that representation. Let's stick to something looking a bit like nature. So this is a slightly more general form of this tiny genetic algorithm than last time. Let's think of these thetas as real numbers. So what happens is that we breed, once again, one at a time. We pick a random one to breed, we rub it out, remove it completely, and we propose a new value, theta tilde k, which is, and how do we do that? Well, with 1 minus the mutation probability, we'll pick any of the other ones, doesn't matter which, and use that, propose that. And with the mutation probability, we have a mutation distribution H. Now, last time I was saying the mutation was either going to be a 0 or 1. This time, so H is a distribution over anything. Let's say real numbers. It could be a normal distribution, it could be, I'm going to give you an example in a moment where it's a uniform distribution between 0 and 1. But it could be considerably more complicated than that. It could be a full multivariate Gaussian distribution. Okay. So I'm not limiting the complexity. These can be complicated things. But when we have a mutation, we have a base distribution, H, which gives us a new element. So this is a rather more powerful, powerful type of mutation than we had before. These kinds of models were proposed for genetics in the 1970s. So, I'm going to go through this. We're going to have a break, and then I'm going to put a few little questions up and make you do a few calculations, and then we'll look at the results. There is one law of teaching, is if you set anybody any form of surprise calculation, this is always wrong. So I should expect not a single person to get anything right. That's perfectly okay. This is situation normal. I think doing maths is very similar to programming. If you're programming away in MATLAB, if 50% of the things you write down are correct, you're doing quite well. Depends on your sort of level of mental threshold for actually sort of typing something onto the screen. Some people are very, very careful and they get 3/4 of it correct. But it's probably slower overall. And it's exactly the same with maths. So, anyway, we're going to do that. Now, once again, we've got this sort of curious idea. Let's suppose, I can suppose in a way, this is just like an urn. This is just like the urn models we had before. The urn model we had before, you have an urn, there's a pot, and you have a certain number of elements in the urn. Before they were black and white balls, now they're, I don't know, they're 2.1, 0.7, 0.86, or whatever. You start off with some starting elements in the urn, and this is really the same urn model. On each time, we pick one of them out, we look at it, we duplicate it, and we put it back in. Because when we resample here, if I'm going to copy, say, θ2 into here, then of course I don't destroy θ2. I, as it were, pick it out of the urn with n-1 things in it, and plonk it back in. And then take a copy and put that back in, and it's got n things in it again. Right. What's the starting thing that we have? The starting thing we have, well, this is a distribution, h. If it's, so we haven't really got an integral number of things here. What we've got in the urn, actually, is we've got, if you like, we've got kind of alpha h in the urn. We've got a little device. This h is called the base distribution. We've got alpha h in there, And so when the first time we go into the urn, we're scrabbling around, we're trying to find something, the urn is nearly empty because it's the first pick. The only thing we can find is the H thing. And imagine this is like a little button. Okay? You hit that button and it pops out an object, distributed according to H. And this whole little button is only of size alpha. Right? And alpha is greater than 0, better be greater than 0, but it could be tiny, it can be like 0.01, or it can be huge, it can be like 100, 1,000. So then, we get our first thing and we plonk it back and it's a ball, like 1.1, like that. Now, the role of alpha now becomes clear, because on the second draw, when we put our hand back in again, our chances of picking up this ball, this is 1, this is are 1, and this is a kind of size alpha. So the probability of picking, say, on second draw from this urn, P of pick ball equals 1 over 1 plus alpha. And if alpha is very, very small here, you hardly ever pick it. And P of pick H equals alpha over 1 plus alpha. I mean, maybe we ought to do it with a sort of arrow, and we start off with a little tiny part of the thing marked, and then we just put up unit things. So then we get so suppose we pick 1.1 again, and we get another ball size 1.1, and on our third pick, it's, well, whatever we do, we'll have 2 balls in there. On the third pick, the chances of, on second draw, on third draw, okay, on the third draw, P of pick H, you can press this little button and get a new value, equals alpha over 2 plus alpha, because there are 2 balls in there which each count as 1, and alpha, which is that. This is a rather elegant process. It's kind of even more elegant than the ball process. This is called the Blackwell McQueen process. It's pretty cool. So that after we've done this for a while, what's interesting is that if, say, H, our base distribution, yesterday we thought of it as just a distribution, it could be either 0 or 1, so it could give you the same kinds of balls that were in the urn already. If we say it's a normal distribution or a uniform distribution, every time we draw something from it, it's a real number. So we're never going to get the same value twice. Okay? So what's going to happen? We keep drawing. Let's get a little bit of intuition. If alpha is small, like 0.1, okay, we plonk our hand in on first draw, and we've got to pick out a ball, so we just scrabble around, but we're only going to find is the alpha H device, which is a special ball with a little button on it, and we pick that up, we squeeze the button, and it gives us a ball, and we plonk it back in. The second time we put our hand in, well, there's a ball there which counts as 1, and the alpha is 0.1. And so, with about 90% probability, we'll pick out the ball again. Right, okay, copy that, 2 balls. And so, we will have really quite a long time before we get another draw from alpha H. Okay? And then we'll get a second value. So, what will tend to happen is that there'll be relatively few values in this worm. There'll be lots of balls with lots and lots of repeated values. If we have, if alpha is big, if alpha is like 10, the situation is reversed. First time you put your hand in, well, you get a ball. There's nothing else you can do. You have a new ball by pressing the alpha button, the alpha H button. Plonk the ball back in. Then the second time you put your hand in, well, you're very likely to find alpha H again, because that's worth 10. The ball's only worth 1. And so you'll get another value, and another value, and another value. So if alpha is quite large, there'll tend to be lots of different values in there. And this alpha is this skill-free parameter which tells you, which is going to control the number of different elements in the earn process. So, if we look at this, this is our tiny GA. We've got 1 minus u times unit for the pick, and then u times h. And we're going to split this over here. Well, not even right. Good heavens. There it was, I just showed all these slides in front of loads of people. There should, of course, be an n minus 1 up here, because we've got n minus 1 balls in there, and n minus 1 plus alpha total probability mass. Okay? Sorry. That's quite wrong. Oops. Because you see, what I want is that n over n minus 1, n minus 1 over n minus 1 plus alpha equals 1 minus u, and alpha over n minus 1 plus alpha equals u. Right? Getting those. So, that's really painful. This slide is wrong. Never mind. That's what it should be. So this is close to 1. As n becomes larger, this is close to 1. As n becomes larger and larger and larger, you will almost inevitably pick out a value that's already in the urn and duplicate it. Whereas your chances of a new value go down as time goes on. But so this distribution of values in the urn is a very odd kind of distribution. There's lots and lots of repeated values, but in the absence of selection, and clearly each one of these values is originally drawn from H, so that it kind of approximates the base distribution. Now, this particular kind of distribution is known as a sample from a Dirichlet process. I'm not going to go into Dirichlet processes because that's somewhat beyond the scope of this course. But this Blackwell-McQueen urn scheme is an extremely intuitive way of constructing a distribution with lots of repeated values, which is kind of closely related to H. If alpha is small, you'll have lots of repeated values. If alpha is large, you'll have lots of different values. And the curious thing is that U is related to N. So alpha, we're just reparameterizing here. Now, the nice thing about this distribution is that this resampling 1 from here is exactly the same algorithm as the breeding algorithm from the tiny genetic algorithm. So they both define Markov chains. In fact, they must define, if we choose alpha like this, then we can, these two Markov chains defined by this breeding process or this resampling process are identical. And there's nothing pathological about these Markov chains. They're perfectly good Markov chains. They're not periodic or anything, so they must have the same equilibrium distribution. So what we've got is, if you like, an explicit, we've got a representation of the distribution, the equilibrium distribution of the breeding algorithm. Let's get a little bit more. Notice how different the different pictures. With the tiny genetic algorithm, we've got this breeding process, which is a Markov chain. So we can start off with anything in the population at all. We can start off with the population being all ones are all zeros, or if our underlying h is, say, a uniform distribution between 0 and 1, we can start off with it all having the value of 0.5. Then we start breeding. And we pick out, and so what do we do? We rub one of the values out, and then we resample it from the urn. So we're rubbing something, our population, if you like, is our urn. So we strike out one of the things in our population, and we resample, and sometimes we'll resample from alpha h. So new values will start to appear. Okay? The mutation with the frequency is this. Now, as you carry on, by chance, some of these new values will take over the population. There'll be a stream of new values appearing occasionally. Often they'll disappear because they'll be rubbed out before they're ever selected again, before they're ever picked. So we have this markup chain. If we have if we do this for long enough, all traces of our initial population will have disappeared, and we'll have reached some equilibrium, a sample from an equilibrium distribution. Okay. The Blackwell-McQueen urn process is a little bit more direct. With that tells us how to start with an empty urn, except for alpha h, and then just in n draws build up a sample from the equilibrium distribution of the population. So we've got a gain now, an identity. Well, now let's put in selection. The first thing we can say, let's develop a little bit of intuition here. If we have a population, these are our simple organisms with only one gene each. One measure of genetic diversity, which geneticists use a lot, is if we take two random members of the population, what's the probability that they're going to be the same, or are they going to be different? So way back in the day, geneticists could identify, say, 10 different versions of a gene in the population, and a measure of diversity is what's the chance that two people have the same version? Well, again, let's develop a little bit of intuition. If, say, 90% of the population have got the same version, got version 1, and 10% have got some other different rare versions, then if you pick two members of the population, then they're both likely, then the first one's going to have the common version with probability 0.9, the second one's going to have the common version with probability 0.9, so the probability they both have the common version is going to be 0.81, and then there's a small probability that they both have the same rare version. So this population is not very diverse. There's one dominant value. If there are 10 different values, roughly equally common in the population, then if you pick two, the chances are the same. It's going to be only like 1 over 10, or less. So, what about this equilibrium distribution? What is alpha doing here? Well, actually, there's a very nice argument. Now, the Blackwell-McQueen urn scheme is exchangeable. That is, the probability of getting a specific set of values doesn't depend on the order in which you draw them. So that you can answer this question, is in equilibrium, what's the probability that two randomly selected organisms are going to have the same version, same gene, they're going to be equal? We can pick up well, let's look at this. Let's put these as draws 1 and 2 in the process. Okay? And draw 1, we got to hit the alpha button. There's nothing else we can do. Draw 2, well, two things can happen. Either draw 2, remember we've got just one ball in there, and we've got the alpha button. So draw 2, we can reach in our hand. With probability 1 over 1 plus alpha, they're going to be the same, because we're going to pick out the ball and duplicate it. And with probability alpha over alpha plus 1, they're going to be different. So when alpha is 1, the probability that they're going to be the same is a half. It's larger than 1, it's going to be greater than a half, it's less than... So alpha is like a dimensionless parameter which tells you the extent of diversity in the population in equilibrium. Would you like me to show you that the black non-queen distribution is exchangeable? Shall we write this about this probability? Maybe I'll get you to do it. Get you to do it after the break. If you've done it, no, if you've done it, you might have got it right by just lashing onto it accidentally, and you'll be happy. If you've got it wrong, it's much better to get it wrong, because then you have to think about why you got it wrong and figure out how to get it right. So making a mistake is actually far more productive. So we'll do that after the break. Now, let's develop some more intuition. Let's put in here some selection. So instead of just accepting every new gene gets bred by this process, let's put in a fitness function. So we breed something, we regard the breeding, whatever we bred, as a proposal. And so what we do is we look at the proposal, and we have a fitness function which gives you a value. It better be, let's say it's greater than 0 because there's no point. And it's less or equal to 1, and that's simply the probability of accepting. And so we generate a proposal and accept it with probability x into k. And what happens is that we now get a joint probability that factorizes as like the probability of with no selection times the probability of the fitness functions. This is there's a kind of at this point, you pause up and say, hang on, how is this obvious? It's one of these cases where you look at it and you say, is this obvious? The answer is it is obvious, but you need to see just how it's obvious. Let's figure out that it's obvious. It's going to be obvious if the... let's suppose that this is our joint distribution. This 1 over z. Have you seen this 1 over z's all over the place yet? I mean, the other way of putting it is it's proportional to, and then there's some normalization factor. Okay? 1 over z. So let's resample 1. Okay? And if this is the equilibrium distribution, it's going to be completely unchanged by resampling. And if it's unchanged, conversely, if it's unchanged by resampling, it's the equilibrium distribution. Because we've got a Markov chain, it's a nice Markov chain, it's only got one equilibrium distribution. Well, let's think about sampling 1. We sample 1. Well, what's the probability? Get it, it's proportional to the probability of generating it, which comes from here, times the probability of accepting it, which is in there. So the equilibrium distribution is unchanged by resampling 1. So the whole thing must factorize like this. I have a slightly longer version of that. Let's develop a little bit more intuition. Okay, the key. Let's go on. I want to. Well, the key simplification. Okay. Yeah, what I want to get to is here. What's the effect of fitness on the distribution? We put in fitness. We're selecting. So I want to know what this does. Let's think about this. If alpha is really big, then if we make alpha big enough, if we make alpha like 1 million in here, then pretty much every time and we've got our n balls, if alpha's 1 million and n is, I don't know, 50, we've got 50 balls in that, And pretty much every time we reach our hand in, this is going to be huge. We're going to hit this. So when we breed, we eliminate a ball from the air, and we hit this button, and we get a new value. So with quite high probability, all the elements of the population will be different. Alpha being very large corresponds to a very large mutation rate. Whenever you breed, it's almost always going to be a mutation. And a mutation is going to be a new draw from a new distribution, from a continuous distribution. It's going to be different from all the other values. So you're going to have different values. And in that case, your marginal distribution of theta, the values of theta you're going to typically find in your population, is simply going to be proportional to the probability of generating that theta under h times the fitness. Let's take the other extreme. Suppose that alpha is really small. If we make alpha small enough, then nearly all the time, this iron's going to have balls which are all the same. So in that case, the probability of the theta has a particular value is going to be proportional to the probability of generating that value times the fitness of that value raised to the n. For intermediate values of alpha, what happens? Well, let's just do it in MATLAB. So here we are. This is what you get. Here I've generated values of alpha, which are between 0 and 1. So, sorry, not alpha. I've generated values of, if you like, theta, x, between 0 and 1. These are all keen value, or allele values. Then I've made the fitness to be just the allele value. So the probability of accepting a value of 0.1 is 0.1, and the probability to accept a value of 0.9 is 0.9. So I have a sort of population of 20. So if we have the yellow graph, which is a population of 30, most of those values are going to be different. Some of them are going to be the same, because it's not a million, but most of them are going to be different. And so what you get is a distribution which is pretty much, it's nearly a straight line. It's slightly biased here because we've got some groups of 2 and a few groups of 3. If on the other hand I take alpha as 0.1, then nearly all elements of the population are the same. If all elements of the population are going to be the same, the fitness, the element you have is this blue graph is nearly proportional to x to the 20th. So x to the 19th. Because most of the time, they're all the same value, and the joint probability has got to be proportional to h of theta. This is also the marginal probability has got to be proportional to h of theta times f of theta to the 20th. Because n is 20 here. So this says something kind of important. That lower mutation rates enhance the effect of selection because you have repeated elements in the population, and equilibrium may take a long time to get to, but the equilibrium is going to be much more sensitive to fitness. If you have high mutation, you're constantly getting new values suggested, which may not be fit at all, and these accumulate in the population by chance, and selection gets rid of these new values. And the selection gets rid of the values at kind of the same rate, they're always being injected, and so you get a curve much more like this. For intermediate values, so very low mutation rates, from a genetic algorithm, are a compromise between sensitivity to fitness, and we've got time to reach equilibrium. Let's look at this. This gives you the group size. This is this gives you the fractions of elements in by group. I mean, in your urn, the urn has 20 balls. They all got different values. What was it? A group is a set of balls with the same value. So when alpha's 30, then the most common, most balls, more slightly over half the balls, have a unique value. We're in a group of 1. And then some of them in groups of 2, 3, and 4. But you never get all 20 balls the same. Because when you're reading, you eliminate a ball, you reach your hand into the urn, and most of the time, you pull out a new value because you hit the alpha button. Does that make sense? Okay. At intermediate values, you get groups of all sizes. And for alpha is 0.1, we can see that nearly all the balls are in groups of size 20, and just occasionally you get a group of size 19 or 18. You might say, hang on a moment, if you get a group of size 19, that must be some group of size 1. Yes, this graph shows the fraction of the balls in a group of size 19. So for every situation where you've got a group of size 19, 19 out of the 20 of the balls are in this group of 19 here, and 1/19 of that number is down here. So you should see a little tiny uptick, but it's times 1/19, so you can't really see it. So I think that was the best way to plot this graph, but maybe I should have plotted it both ways, both in numbers of groups and the fractions of balls in a given group size, to make that right. Okay? So this gives us a little bit of intuition. Now, you might say, well, surely we should be able to get nice analytical expressions for the marginal distribution for intermediate values of alpha. We've got an extraordinarily simple and elegant generative process, this Blackburn and Queen Urn scheme. It really couldn't be more elegant than that. It's deceptively elegant, but it couldn't be more elegant. Surely, and my fitness function is again one of the simplest things I could have produced. Surely there must be analytical expressions. Well, I asked Ui Te, who is kind of expert on this kind of thing. He said, well, actually, Chris, for intermediate values, you can't usually get analytical expressions, and that's why people do MCMC. That's why people do Markov chain Monte Carlo, because you can't get analytical expressions. But I didn't quite believe him, so I plotted log plots and stuff. Nothing gives you straight lines. So maybe there are nice analytical expressions for this, but I just don't know. Now, let's put it all together. Okay. So far I've been talking about tiny genetic algorithms. Let's have slightly larger genetic algorithms. Let's have each column is independently a tiny genetic algorithm. Okay. And the genetic, biologically inspired breeding is to do a whole row at a time. That is, we eliminate an organism, so we zap an entire row of these. Then the breeding process is to generate, propose a value here, propose a value there, propose a value here, propose a value there, and then accept it all according to the fitness function. So that we're thinking of it as a set of different tiny genetic algorithms are linked by one fitness function. So that's the Gibbs breeding with selection. You can also have a column. You can do an individual. The joy of so this model still has the property that the probability of this entire tableau, we've got a whole table of random variables here. This is our whole population now. We're describing our population as a whole rectangular table full of random variables. Well, the joint probability of that is proportional to the probability of each of the column distributions under no selection times the probability of all the fitnesses. Times all the fitnesses. That's kind of, this is cool because it means that we don't have to breed in this biological way. We can do other things. We can do one allele at a time. And so that's the biological idea. There are also ways of doing a column at a time. We can have other algorithms, which, so given a probability distribution defined as a factor graph, you can define lots of Markov chains that converge to that same equilibrium probability distribution. Okay? So all of these things will find you the same distribution in the end, but some of them may do it much faster than others. Right. So this is just a breeding algorithm, which I think I'll leave you with. Yeah, let's leave you with this breeding. Right. So I want to have a break at this point because it's a good idea. So I'll see you back here during the time it takes to get a cup of coffee, and then we'll do some calculations, which it's much more productive to get wrong. And then we'll, after doing that, about these distributions, I will, in the third part, I'll talk about a quite different approach to looking at genetic variety in populations, which I think also gives a lot of insight. It's also beautifully simple and also gives a lot of insight. Okay. Well, I'm going to be back here in 10 minutes.